Hello, everyone, and welcome to today's webinar, Building Panels for Flow Cytometry, Key Steps to Success. I'm Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Cytek Biosciences. To learn more, please visit cytekbio.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Laura Johnston, Technical Application Specialist at Cytek Biosciences. Laura, welcome. You may now begin your presentation.
Thank you. Okay, so I am going to cover a lot of panel design best practices today. And I want to start out by saying or making the point that pretty much everything I talk about today, you can apply to all platforms and panels of all sizes. So it doesn't matter if you're using a conventional cytometer or a full spectrum cytometer or whatever panel size you are using, I should be able to give you some really good information today. Now, of course, depending on the scenario that you're in, you might want to use slightly different strategies. So as I go through the talk today and I talk about these panel design best practices, I'm going to do so using 3 different scenarios as examples. So the first scenario is your smaller, simple panels. And really, this is the basic information that everyone should find useful. The second scenario is for those of you who may be interested in doing something that's a little bit larger, a little bit more complex. I'm going to give you some strategies to apply to that specific scenario. And then the third situation is for those of you who may be running a panel on cells that have high autofluorescence. I'll talk about exactly what that is, how you figure out if you're in that situation, and some tips for that specific scenario. So remember these 3 scenarios. I'm going to go through them as I go through the talk, and I'll call these back. Okay, so for the workflow and the main structure of this talk, I'm going to be talking about the panel design process in 4 steps, and it really should be informed. So to start out, we are going to gather information. So the first thing we want to gather information about is the biology. We want to know our scientific question and what populations we are interested in investigating. The second step is we're going to gather information about the fluorochromes, and that's going to come down to our cytometer configuration, and we'll also talk about some characteristics of the fluorochromes so that you can combine them together in a panel. The third step is where it gets interesting. We're going to start pairing the markers to the fluorochromes to design our actual panel. Now, this is not the last step. There is a step after that, which we're calling the optimization. And this is where you're actually going to be able to determine if your panel is successful or not. So we're going to walk through all 4 of these steps using the examples that I outlined on the previous slide. And let's start with our first step, which is the biology. We're going to talk about how you go about selecting markers, the level of expression, and thinking about co-expression. So starting out, we really want to have it clear in our mind. What is our scientific question? What are we trying to address that will help us understand what populations we are interested in looking at with our panel design? Now, if you are looking at immune markers, you could think about these different loose groups of markers. This is just an example strategy. You don't necessarily have to use this, but it can be helpful. So our first group is lineage markers. Let's say you're interested in T cells. So the lineage marker for T cells is CD3. We would want to include that in our panel. The second marker, or the second group, is additional markers. So maybe we're interested in subsetting T cells further. So if we want to include CD4 and CD8 to subset our T cells, those would loosely go into the additional markers group. If you're interested in exclusion markers, you may not include these, but if you would like to exclude certain subtypes from your panel, let's say I only want to look at T cells and I don't want to look at B cells, I may include a B cell marker as an exclusion marker so that I can remove those from my analysis. And then other markers is just dependent on your scientific question. So maybe you're interested in cytokine receptors or activating receptors, whatever you may have. Now, having these loose groups can be useful when you're thinking about the next step, which is co-expression. So for lineage markers, oftentimes one lineage marker is mutually exclusive from another lineage marker. So our CD3 is not going to be co-expressed on a totally different cell type like a neutrophil. Our additional markers, our CD4 and CD8, oftentimes those are co-expressed with our lineage markers. So we know that CD4 and CD8 are going to be co-expressed with CD3. And then our exclusion markers, we have specifically put those in so that they're going to remove cells that we're not interested in. So they're not going to be co-expressed with any markers that are on our cells of interest. That's one way to start thinking about co-expression. There are a few other ways. So these are some visual methods. On the left here, we have a cell lineage tree, and we can see at the bottom of that, we have CD3 that's being used to define our T cells, but the CD3 is not on our NK cells. So that's another way to think about things. Or on the right side, we have a gating strategy. Now you can do a rough gating strategy like this, where you just sort of loosely plan out how you're going to gate your populations of interest, or you may actually go into publications and find some real data that will actually show you populations in these plots. But we can see, for example, CD56 and CD16 on the second plot, we're looking at a few different populations, even a double positive population with these markers. With the list of markers, we also want to start thinking about reagent availability. You're probably going to be purchasing antibodies from various vendors, and so we want to know what is available so we can put those into our panel. We also want to be thinking about clones. Not all clones behave similarly, so it's important to learn about what clones are available and what people are using in publications. And then experimental conditions. If you're doing anything to your cells, maybe if you're stimulating them, thinking about how your markers are going to be affected by those experimental conditions. So there's a lot of research, or there's a lot of information out there. I would encourage you to do some research. As an example, in the green box, we have what you can find on the reagent page for cytekbio.com. You'll find information about clones, fluorophores. Oftentimes vendors will give you information about the markers as well. So that's one starting place that you can go to look for information about reagents. You can also look at publications. So if you're interested in NK cells, for example, maybe you find this publication and you can learn about the markers, how they're defining the NK populations. You can pull information about clones from papers. So it's really good to do some research on the field that you're looking at. Another option I want to highlight is in the journal Cytometry Part A. There are a subset of papers called OMIPs, that stands for Optimized Multicolor Immunofluorescence Panels. And these are really focused on panels. People have spent a lot of time optimizing these panels, and they will give you all the information in these papers. So there's really a ton of good information, and this is really a great starting point for you to look through these various OMIPs. And then a last reference that I will point out is this paper in Frontiers in Immunology. If you're interested in human markers, specifically human immune markers, there is this paper that gives you a ton of information about the CD markers numbered 1 through 100. So that's another great resource there. But again, do your research and figure out what is available in your field. When you have your list of markers, you also need to start categorizing your markers. That's going to be very critical when we get to the step where we're pairing to our markers to our fluorochromes. The categories come from this paper that is referenced in the lower right-hand corner by Monkey and Roederer. The categories are primary, secondary, tertiary. So a primary marker is very high density. It typically has an on-off pattern. So you can see in that data plot there, the cells either have the marker or they don't, and there's no in between here. The secondary marker, however, you could describe it as its intermediate density, or maybe it has a continuous expression. So we have both low density and high density antigens, and we want to be able to resolve all of that expression. And then our tertiary markers are our low density markers, or potentially our unknown expression. Now, a note about unknown: if you yourself do not know what the expression is, do some research, see if somebody out there knows what it is. But if really nobody knows, maybe that's the whole point that you're doing this experiment, then you can categorize it as a tertiary marker. And so it's important to understand these categories because when you get to pairing your markers with fluorochromes, you need to make sure that the tertiary markers specifically get paired with very bright fluorochromes, because if you pair a tertiary marker with a dim fluorochrome, you will not be able to optimally resolve it. So moving on to the fluorochromes, let's talk about information we need to learn about the fluorochromes, starting out with the instrument, as well as some criteria for selecting the fluorochromes. For the instrument, it's really critical that you understand the specific instrument that you're going to be using for this panel. Particularly, there are 2 points that we want to understand. The first has to do with the lasers. So what lasers are available to excite the fluorochromes that you're going to be selecting. Typically, the information we want to know there is the wavelengths of each each laser. And then the second question has to do with how the instrument is going to be detecting these fluorochromes. And so this is probably the only point where I'm going to mention a difference between a conventional cytometer and a spectral cytometer, because those are configured a little bit differently. So our example image on the right there is a 5-laser Aurora, which is a full spectrum cytometer. And the way that this instrument is configured is we have a detector array that matches each laser. So 5 lasers have 5 matching detector arrays. And ultimately, there's going to be a grand total of 64 detectors in this system. And all of these detectors are going to be utilized to pick up the fluorophore emission for all of the fluorochromes. So actually, if you're using a spectral system, it becomes incredibly easy because all we need to know is what lasers are on board, because we know there's a matching detector module. And that's really all the information that is needed. It's also great to know that the Cytek instruments are very standardized configuration. And so all of the tools I'm about to go through will be applicable to all of the Cytek instruments. For a conventional system, it's a teeny bit harder. You're going to need to know exactly what your system is configured as. So you need to know how many detectors are present, exactly what wavelengths they can detect. So another way to look at this, here is a 5-laser Cytek Aurora again. The detectors, all 64 of them, are on the x-axis here. And so you can see the signal that we're getting from this particular fluorophore. It's going to be picked up by all 64 detectors. Whereas if we have a conventional system, we're going to be instead assigning a single detector, specifically where the emission maxima is. And so on a conventional system, your data would actually look like this. So it's just a single detector. Now let's go into the criteria for selecting fluorochromes. There are 3 criteria that we're going to go through. The first one is uniqueness. So we want to make sure we're selecting fluorochromes that can be distinguished from each other. The second is brightness. As I mentioned, we want to pair the brightness of the fluorochrome based on the antigen density. And the third one is spread, which is a slightly more advanced topic that I will explain in detail and point out those of you who need to pay more close attention to the spread. So in terms of the uniqueness, if you're using a Cytek system, we try to make this super easy for you. And the way we do that is we give you this value called the similarity index. So that's a value that's between 0 and 1, and it compares 2 fluorochromes. Let's go through some examples. So we have BUV395 in green and Seafluor R720 in purple. If you look at that second plot in the middle there, you can see there's no overlap between these 2 fluorochromes. So because there's no overlap, we have a similarity index of 0. It means the spectra are completely unique. And in our data plot, we can see that we can define these populations really nicely with these fluorochromes. If we take the second example, now we're using APC and Alexafluor 647. In this case, we have a lot more overlap and our similarity index is 0.9. So this value tells us that we can use these in a panel together on a Cytek system. However, I will note that these on a conventional system will typically be assigned to the exact same detector. So I would not recommend putting these together in the same panel on a conventional system. But for a spectral system, we can do that, and you can see the data plot below looks very nice. Then we have our third example, which is BV786 and Superbright 780. These have a similarity index of 1. And you can see that our data plot below there does not look very nice. We can't resolve our populations. And so we should not be using these fluorochromes together in a panel because we cannot distinguish them. So using the guidelines at the top, if the similarity index is 0.99 or 1, then don't put them in a panel together. If it's between 0.94 and 0.98, we would recommend doing some testing. It's going to depend on the markers that you select for those fluorochromes as to whether or not you can resolve them. But anything below that, you should be able to distinguish. And unfortunately, for a conventional system, I don't have a matching metric. You're just going to have to understand your specific configuration and what fluorochromes you can detect. Now for the similarity index, we typically show you in a grid like this. So that triangular shaped grid there has all the similarity indices inside of it. But you may want to think about a different metric. So what about all of the colors combined together? So the similarity index only compares 2 fluorochromes. So in this example, we have 14 colors. And if we look overall, we have a complexity index, a different metric that will describe all 14 of them combined. And in this case, it's 7.89. So the complexity index gives us an overall measure of uniqueness. And unlike the similarity index, it doesn't have a hard cutoff value. The complexity index instead just gives us a rough estimate of if it's lower, then it generally means that is an easier combination of dyes to work with. So to give you a point of reference, our 14 color kit is 7.89. If you have a smaller panel, your complexity index may be something like 4.02. But if you go much larger, like our 40 color panel, the complexity index is 53.7. I don't want you to get too hung up on the precise values, just that there's a relative comparison of lower values are easier to work with, higher values are harder to work with. And I'll give you some examples of exactly why that is. So the second metric is brightness. And in order to quantify brightness, we generally use this metric stain index. So the calculation is shown there. It basically just tells us how well we can separate a positive population from a negative population. And so ultimately, what's most important to understand from the stain index calculation is that there are multiple factors that impact it. The actual stain index itself, as well as the signal intensity. So the factors are antigen expression, fluorochrome brightness, instrument sensitivity or setup, and antibody performance. So if you change any of these factors, it could impact what your ultimate signal intensity is. So in order to come up with a tool for panel design to figure out what is the brightness of the fluorochrome, we need to basically fix all of these factors and just change the fluorochrome brightness. And ultimately, that's how we came up with this tool, where we ranked over 100 fluorochromes on a 5-laser system. And so if you use this chart, it will basically tell you that if you're looking at a fluorochrome on the right side of the chart, then that is generally a very bright fluorochrome. And if you're looking at one on the left side of the chart, that's a very dim fluorochrome. So use this as a general estimate. It is a continuous scale. You can see, even though we've boxed it into 4 categories, it's a continuous value. And these can be helpful as you're designing your panel. Okay, let's get into some actual examples. I'm going to be going through those scenarios that I talked about in the beginning. So I put a little icon in the upper right-hand corner to designate which scenario I'm talking about. So in this case, we're talking about scenario 1, which is our small, simple panels. So everyone's going to start out with a small panel, and then some of you might be building it bigger. So to start out, I would recommend that your simplest approach for selecting a group of fluorochromes is to pick fluorophores that are both excited by different lasers and have different emission maxima. So in this example that I have here, I've selected 5 fluorochromes for a 5-laser system. They're all excited by different lasers, and we can see the emission maximum listed at the top there ranges from 395 nanometers to 774 nanometers. However, if I was instead to select this combination, where now these 5 fluorochromes are all excited by the exact same laser, this is certainly not an impossible panel to use. You could find success with this panel. However, it is probably going to be just a little bit more challenging to design this panel compared to the panel on the left. You just have to use a little bit more caution, you have to be a little more thoughtful. That's a better word, if you're using the combination on the right. So I would say it's easier to start out on the left. And then as you build your panel larger and larger, we want to make sure that we're continuing to keep everything spread out. We want to minimize our similarity and complexity indices. So moving to our next example, as you notice in the upper right-hand corner, we've now switched to the second example with a larger complex panel, let's look at a 15-color panel. I'm going to call this one panel A. And in this case, I've put our fluorochromes into our panel matrix. So let me describe the panel matrix so you can figure out how we can use this panel matrix to assess our panel. So the panel matrix is set up with the columns designating lasers, and that's going to position our fluorochrome based on the primary exciting laser. So the first column is UV, and so inside of that column we have BUV 396, which is excited primarily by the UV laser, whereas on the right side we have the red laser, and in that column we have CFluor R659, which is primarily excited by the red laser. The rows are going to designate the approximate emission. And so the top row is 395 nanometers and the bottom row is 800 nanometers. And so based on that information, we place fluorophores in the matrix. And so we can see just visually that everything is pretty well spaced out, spread relatively evenly across all of the lasers there. And in our panel A, we can see in the top, the complexity index is 4.1. So let's switch this to panel B, which still is 15 colors, but now the complexity index is 12.96. And we'll notice that our fluorochromes are centralized all in the center of this panel matrix as opposed to being spread out. Even if we focus on the blue laser, we'll see that we have two scenarios where we have a cluster of 3 fluorochromes stacked on top of each other, as well as if we look in the same row, we have 3 fluorochromes in the same row, indicating that they all have the same approximate emission maxima, even though they are excited by separate lasers. So if you have fluorochromes placed in the panel matrix like this can indicate that your panel is a little bit more challenging to work with. This is certainly not an impossible panel to use, but you're going to be a little bit more thoughtful when you design your panel. So why is panel B more difficult than panel A? It comes to our third topic, which is spillover spreading error, which we often just abbreviate to spread. So spread has to do with spectral overlap, and I'm going to walk you through what that means, and then we're going to revisit panel A and panel B. So in this example, we're going to use PE-Sci5 and APC as our example fluorochromes. This is run on a spectral instrument, but honestly, you can even pretend this is a conventional instrument. So we have 3 rectangles there. You can imagine that those are 3 separate detectors on a conventional instrument. So the blue rectangle in the middle, we would typically on a conventional system assign that to PE-Sci5 because it captures the emission maxima of PE-Sci5, whereas the red rectangle would be assigned to APC because it captures the emission maxima of APC. But you notice what's different between the red box and the blue box is that in the red box, we are also seeing PE-Sci5 signal in addition to APC signal. So if I said this in a sentence, I would say that PE-Sci5 is spilling into the detector assigned to APC. Now, this is before we've calculated any compensation or unmixing. So if we actually go ahead and calculate that, that's where we would start to visualize this spillover spreading error. And so if we look at the APC positive population, we see that there's minimal spread. Our positive population is nice, round, compact population, whereas the PE-Sci5 positive population is much more spread out. So this is the spreading error that we're talking about. And the result, or the reason for this, is because the PE-Sci5 is spilling into the APC. Now, it is important to understand that spread can be impacted by multiple factors. So I mentioned one of those factors, which is spectral overlap. And that's why selecting fluors that are unique, have a low complexity index that will help minimize the amount of spectral overlap. But there's also additional factors. So the brightness of the impacting fluor, as well as the antigen density. So those 2 things will impact the overall final intensity of your positive population. And as you can see in that third example, as that intensity gets brighter and brighter, you can see that there is more spread. So some people call this the trumpet effect or the umbrella effect, because this is a pretty classical pattern of spread. Now, one important thing you will note that I did not say anything about a conventional system or a spectral system impacting spread. So that is not going to change your spread, especially if you have a panel that is working well on a conventional system and you decide to use that exact same panel on a spectral system. you absolutely will not get more spread. At worst, you would get the exact same amount of spread as you did on the conventional system, or depending on the panel and the systems you're using, maybe you might even get a little bit less spread on the spectral system. Now, how do we assess this for panel design? We use a tool called the SIR matrix. This is something you could calculate for your actual panel, but what we've done here is giving you a tool to assess the worst case scenario. So we ran CD4 on all these fluorochromes listed here and calculated the SIR matrix to assess the worst potential spread that you could get, or a pretty bad case of it. If you want to do this on your system, you would either have to figure out if anyone has done this for your specific configuration, or you can make it yourself. But the way you would read this matrix is the die in the row impacts the die in the column. So let's choose a row and see how our data gets impacted. So our first row is seafluor B515, and we're going to compare that. How is that going to impact the resolution of the fluorochrome, or the marker that we assigned to the fluorochrome, in the column? So if we look at our data here, because that box is white, so there's no impact, and we can definitely see that in the data, we're not seeing any spread of seafluor B515 into seafluor V420. But if we go along the row to a place where there's a red box, now we're seeing that there is spread into seafluor B532, which is the right plot there. So in terms of panel design, we need to think about what is the antigen density and how we're pairing our markers to these fluorochromes that are on the y-axis of these two plots here. So if you have a marker that is placed where that green circle is, then you should be able to resolve your data. And notice how for the plot on the right, we need a little bit more space between the positive and negative population to account for that spread. But if we were to use a low density antigen on that seafluor B532, then we risk not being able to properly resolve that population because of the spreading error. So back to our panel A and panel B. Why is panel B more challenging? It's because panel B has more spread. And if we look at the SIR matrix compared to panel A, we can see the SIR matrix for panel B has not only more boxes that are red, but within the boxes, the actual values are higher. So that's why we have to use more caution when we design panel B, but it's certainly not completely impossible to do. Okay, now we're going to switch again into our third scenario, which is cells with high autofluorescence. You can see our little icon in the upper right-hand corner is now switched to the number 3. So how do you figure out if you're in this scenario or not? Well, if you have no idea, I would say start out, before you do any panel design at all, take some unstained cells and run them on your cytometer. And that will give you an idea of how much autofluorescence you have. This is especially easy to do on a Cytek system. If you're using a conventional system, you are going to need some optimized settings to be able to make this call. So in our 3 examples here on the top, we have red blood cells. Intensity in these plots is on the y-axis. So we can tell that the intensity of the unstained red blood cells is pretty low. In the middle plot, we have lymphocytes. So those generally are not too high. They're kind of on the lower end. But our third example, alveolar macrophages, those are very highly autofluorescent. And if I gave these a value, I would generally say that these alveolar macrophages are at least sitting at 10 to the 4. And there is no stain in these at all. So we do need to pay attention to how we design our panel if we are in this high autofluorescence situation. And another note, if you are going to run your unstained cells prior to doing any panel design, I would certainly recommend that you record a ton of events. So if you're planning on running 200,000 events in your final sample, your multicolor sample, run about that many in your unstained to make sure that you can fully assess all of the autofluorescence in your sample and there aren't any rare populations that will surprise you later. So what do you do if you're in this high autofluorescence situation? If you're in the low autofluorescence situation, like the lymphocytes, you're going to ignore what I say right now. This is only for those of you who are in that high autofluorescence situation. The fluorochromes listed here are ones that typically have a emission maxima that overlaps in regions where there is a lot of autofluorescence. So you have to be aware that there's a potential for you to have lower resolution with these particular fluorochromes, and you may need to be strategic about how you put these in your panel. And again, depending on your panel, if you have a very small panel and you are able to not include a fluorochrome, let's say BV421 tends to be a pretty difficult one. So maybe you just don't include BV5, or sorry, did I say BV421? I meant BV510. So BV510, you might just not put that in your panel and leave that channel open. That's one option if you have the availability to do that. But if you want to include that, then in the next section, I'll talk about some strategies you can use to include these fluorochromes. So at this point, we have gone through gathering a ton of information about both the markers and the fluorochromes. Now, with that information, I'm going to talk about how you would go about designing a panel. First, I'm going to go through a tool that we provide to you at Cytek. It's in Cytek Cloud. It's called Panel Builder. And then I will actually give you some examples for designing some panels in these various scenarios that we have been going through. So for Cytek Cloud, it's a tool that we have recently released. There's a ton of really great information in there. It has all of the tools that I've already mentioned up to this point. It also has all of our kits listed. It has all of the OMIPs are listed in there as well. So if you want a starting place, you can start from a kit. You can start from an OMIP. That's an option for you. But if you want to sign up for a free account, you can just go to cloud.cytekbio.com and create your own account. Let me show you how the panel builder works. So this is a screenshot of the main area that you will use to design your panel using the best practices that I'm going through today in this talk. At the top, we have our Spectra viewer. In this main portion here, we have our panel matrix. And I do want to point out that in the panel matrix, the stain index is built in. So there's going to be a colored bar underneath each fluorochrome. And that colored bar is going to indicate the brightness of the fluorochrome on the system that you have selected for your panel. So if you choose a 5-laser system, it will show you the brightness for a 5-laser system. You also will get a bar underneath the marker name separately from the bar underneath the fluorochrome. And the one under the marker is something that you can manually set yourself to help you keep in mind what is the antigen density of that particular marker. We also have the similarity index that's going to calculate based on the fluorochromes that you've put into your panel matrix, as well as the stain index reduction matrix to give you a sense for the worst case scenario of spread. And that's going to calculate specifically based on the fluorochromes that you have in the panel matrix, which is really, really awesome. And then to design your panel, you have a list on the left there. And so you can type in a fluorochrome. When you do that, you will get all the available options. If the option does have a little Cytek icon, that little rainbow circle-y thing, that will indicate that that particular antibody is actually in our reagent catalog or one of our partnership reagent catalogs. You can also interact with the panel matrix as well. And so any information that you enter will show up both on the list on the left as well as in the panel matrix. So this is a really handy tool to use all of these best practices to design your panel. When you're done with your panel, you can export that as a PDF. You can send it to a friend in Cytek Cloud, or you can also use the Create Experiment button, which will create an experiment template that you can load onto any Cytek system. And basically, you will just be able to show up at the cytometer and immediately start running your samples. So let's get into our situations again and go through some example panel design. So moving back to our simple scenario where we have a smaller panel, let's go through the process of assigning your fluorochromes to your markers. So we're going to start out with our tertiary antigens. Recall that I said it's incredibly important to pair your tertiary antigens with the brightest fluorochrome that you have available. Oftentimes, these tertiary antigens have some limited reagent availability as well, so it's good to place these ones first. Then you're going to work your way down the line, so we're going to move to the secondary antigens, and those are going to go to any bright dye that you still have available or any mid-bright dyes. And then primary antigens can go to any remaining dyes. In a smaller scenario, it's not quite as critical where you place these, but oftentimes the dim dyes work well. But specifically for smaller panels, I probably am not going to be too concerned if you pair it with something that's a little bit brighter. Now let's use this example. We're going to design a 7-color panel on a 4-laser Cytek Aurora. So we're going to start out by classifying our antigens. On the top there, we have CD25. We can see that the positive signal is pretty low. So we're going to categorize this one as a tertiary antigen. The middle row, we can see that they either have intermediate density like CD127, or there's a range that we're trying to resolve both low and high density antigens. So we're going to call those ones secondary antigens. And then the third row has a very clear on-off pattern. So we're going to call those ones our primary antigens. Now we can use a gating strategy if we want. It's always a great idea to start out with a gating strategy, but for the smaller panels, it's not going to be incredibly critical for us to pay attention to co-expression. So when we design our panel, we're going to start with the tertiaries. We're going to assign that to the brightest fluor available. So I'm going to select PE for that. Then we're going to move down the list to the secondary antigens. We're going to assign mid-bright fluors for that. So we're going to go with these 3 here. You'll notice that we have different colors in those bars. So the purple indicates that that's primarily excited by the violet laser. And then the actual intensity, I guess, of that bar is going to indicate the brightness of that fluorochrome. So you can see that BV421, the second one, is not as bright as PE. PE is our maximum brightness, but in general, we still think of BV421 and APC as pretty bright fluorochromes. Then we're going to move to the primary antigens, and we are going to pair those with any mid or dim fluorochromes. And so those are the ones we select. And then the last thing we do is we're going to select a viability dye. Strongly recommend that you put a viability dye in any panel. And typically those can go anywhere you want as long as they're still unique from your fluorochromes that you're trying to look at your markers with. Unless you want to use a dump channel, there are ways to combine a viability dye and a dump channel, which means the dump channel is those exclusion markers that you are removing cells from your analysis. So anything you're not interested in. Now, if you want to do a quick check of our panel design, we can see that there are 2 fluorochromes that are primarily excited by the violet laser, 2 excited by the red laser, 2 excited by the blue laser, and 1 excited by the yellow-green laser. So based off of that, everything's pretty well spread out. I've done a pretty good job of selecting my fluorochromes, and I'm not going to pay more attention than that. It's pretty straightforward when you have a small panel. Of course, if you want to do something that's a little bit larger, then we need to be a little bit more cautious in our design. So we're going to go through the same steps, but I'm going to add some things for you to think about. So our first step is going to be exactly the same. We're going to pair our tertiary antigens to the brightest fluorochrome available. Our secondary antigens are where things change. We're going to start to need to pay attention to co-expression. The more complicated your panel is, the larger it is, the more critical it is for you to pay attention to co-expression. So if there is no co-expression, then you can place your antigen wherever you want. You can use any dye that's still available. However, if you do have co-expression, that's where we need to be careful. I would refer to the SIR matrix, and we want to make sure we are minimizing any impact of spread. And then finally, we have our primary antigens. This is where it's much more critical to pair these with dim fluorochromes. That is a strategy we use to minimize spread. So let's look at our example now with a 19-color panel, so a lot larger than our first example of 7 colors. We have a few tertiary antigens. We have a lot of secondary antigens, a couple primaries, and then our last case is special. So you'll notice we actually have 2 categories there. So we have CD4 and CD3, which are listed both as primary and tertiary. And this is because we're using a stimulation condition. So in the stimulated condition, we have markers that are going to be internalized, so they become tertiary as they get stimulated. So we need to pay attention to that in our panel design. So using our panel matrix, we have selected fluorochromes. We're going to start out with our tertiary antigens. We're going to place interferon gamma, TNF alpha, and CD127 on bright fluorochromes. Remember how we have the bar underneath the fluorochrome indicating the bright fluorochromes, so PE, APC, and APC R700. Then we want to pair our special two fluorochromes, the CD4 and CD3, to accommodate those low density antigens. It's really important to accommodate the tertiary antigens. So we're going to pair very bright fluorochromes. Then moving to the secondary antigens, we are going to, again, just move down the list in brightness and pair those secondary antigens with any remaining bright fluorochromes or any mid-bright dyes that we have. And as we go through and place those secondary antigens, we're going to refer to the SIR matrix and minimize spread. So I'm going to go through one of them, but you would do this for every single secondary marker that you are placing. So we're going to do the example with CCR7. I've placed that on BV421. So we're going to look in the row for that. And we're going to look across the row for BV421, and we're going to compare it to the tertiary markers that we have already placed. And we can see that the box doesn't have any value in it. So the value is really 0. And so this is good because our CCR7 is not going to impact the resolution of any of the tertiary markers that we have placed. So we like this placement here. Moving to the next step, we are going to place our primary antigens. So we have the 3 primary antigens in orange. We have put those on dim fluorochromes, BUV 615, PerCP, and PerCP Bio 700. And so again, with the primary antigens, we are going to want to still assess the spread and see if there's any issues with the placement that we have selected. So going back to our SIR matrix, I'm going to check it for CD45. We've decided to put that on PerCP. So looking across the row for PerCP, we're going to see where is there a value, and it turns out we have a value of 50 for the column for PerCP Bio 700, which we've placed with TCR-V delta 1. So there's a couple things to think about when you have a value. So first, is it co-expressed? Is CD45 co-expressed with TCR V delta 1? And in this case, it is co-expressed. So we are going to need to pay attention to this value. The second is, what is TCR V delta 1? It's a primary marker. So that tells us we have really good separation between our positive and negative populations. And then it's pretty highly expressed. So that's another consideration to take in. And then also, what's the value in the box? So the value there is 50. This value is a percentage, so it's out of 100%. So 50% out of 100% is not the highest value. It's like an intermediate value. So given that, even though it's co-expressed, it's not the highest value, it's 50%, and our marker that's receiving the spread, TCR-V delta 1, is a primary marker. So because there's good separation, it should be able to handle a little bit of spread coming from CD45. And so based on these, I'm going to take a chance that I probably can still resolve TCR-V delta 1. And so I'm going to move on to the next step, which is assigning our viability dye. Again, as long as that doesn't impact our fluorochromes, it's unique from the fluorochromes, I can stick that anywhere. So I'm going to go with live dead blue. Now, why can I stick that anywhere? Because you might look at the SIR matrix and see that there are some concerning values in this. There's some pretty high values in that row. But keep in mind that with your viability dye, the positive cells, the dead cells, are going to be fully removed from your analysis. So assuming you titrated that properly, everything that's positive is going to get removed. And therefore, we can basically just ignore this entire row because anything that is positive for the viability dye is not going to be co-expressed with any cells that you're actually interested in looking at. So we've designed our 19 marker panel, and the last thing we want to do is check the whole matrix and see how we did. I'm going to go through and look for any sort of high values. So I'm going to pull out 4 values here are kind of on the high side, and I would want to think through, am I concerned about being able to resolve any of these markers, or do I think that I've done a good job? So in this case, I'm just going to go with it, and I'm going to see how this panel looks on some actual real cells. But if you decide that you are concerned about your resolution of markers, you may go back and rearrange some markers. But don't forget, there is another step after this. But before we get to that step, I do want to go through our last scenario, which is strategies if you have highly autofluorescent cells. And so I called out earlier that BV510 was a fluorochrome that oftentimes is difficult to use when you have high autofluorescence. So our fluorochrome for BV510 is on the right there, and on the left there I have 2 signatures for unstained cells, I have the neutrophils on the top, which are on the brighter side, and the lymphocytes on the bottom, which are on the lower side. So if you have the situation that you're trying to use a highly autofluorescent cell type like neutrophils, and you overlay the BV510 on top of that, you can see how easy it is to get the same intensity between the BV510 positive cells and the neutrophils. So in order to be able to resolve a neutrophil marker with BV510, we have to keep in mind that that positive signal needs to be higher than the actual background. So in this case, we're saying that the background is 10 to the 4. It's going to depend on what your tissue is. And there's some other examples listed there in the teal box of fluorochromes that are have similar issues to BV510. However, if we put BV510 on the lymphocytes, you can see that now, because the lymphocyte autofluorescence is lower, it's a little easier to separate the BV510 positives from the lymphocyte autofluorescent background. So a different strategy you could use is if you happen to have a tissue like whole blood in this case that has both neutrophils and lymphocytes in it, you could strategically pair the problematic fluorochromes like BV510 with a lymphocyte marker, because the lymphocyte marker has the lower autofluorescence compared to the neutrophils. Now, viability dyes with highly autofluorescent samples is a little bit more challenging. So in the other situations, I said stick the viability dye wherever you want. In this case, there is some strategy. So on the left side, we have our unstained cells. On the right side, we have 3 different viability dyes. And if I overlay the unstained cells and the viability dyes, notice how I put a little purple arrow to distinguish the emission maxima of the viability dye compared to the unstained cells. You'll see that in the bottom plot, you get the most separation, because dyes that are excited by the red laser are probably going to be the easiest to use in terms of viability, because oftentimes the red laser doesn't have a lot of autofluorescence, so you get the best separation. So that would be my recommendation. If you have a highly autofluorescent sample, use a viability dye that's excited by the red laser. Now we can move on to our final step, which is the optimization. This is where we actually determine if your panel is successful or not. Now, how do we do that? We're going to have to run an experiment and get some data, because we need to figure out if we can actually resolve all of our populations and markers of interest. If we can, our panel is successful. If we cannot, then we may need to adjust our panel. So I would recommend doing something that I call a panel optimization experiment. But before we can get to that, we actually need to go through some readiness criteria for that panel optimization experiment. So you need to start out by titrating your antibodies. You're going to be in a single color scenario while you titrate your antibodies, and that's going to allow you to confirm that you have optimal resolution in the single color scenario. So it's a very critical step. Then you're going to also need to figure out how you create high quality single color reference controls, or maybe you call them compensation controls. Whatever single stained controls, you need to follow the best practices for those. And that will allow you to calculate accurate unmixing. So the second and third points kind of go hand in hand. Ultimately, you need to calculate accurate unmixing and compensation, and you need to have that all dealt with before you can actually do a analysis of your panel optimization experiment. I also want to talk about how you would actually set up your panel optimization experiment. The design for it is maybe a tiny bit different than you're used to. So the first thing you want to do is you want to make sure that you're bringing your single color reference controls, or if you want to call them compensation controls, basically whatever you're using to calculate accurate unmixing or compensation, you need to bring those so that you can calculate the unmixing and compensation. Doesn't matter what it is. It could be beads, could be cells, just as long as it gets you accurate unmixing and compensation. The second thing is we want to combine those markers, our panel, into one tube. So you're going to bring in a multicolor sample. I would say ideally you want to run this on your final sample type, but there may be situations where you don't do that. So if you want to use a healthy donor instead of one of your patient samples, just know that if you use an alternative, your panel might behave a little bit differently, especially if there's a drastic difference in the marker antigen density between a patient and a healthy donor. So proceed with caution. And the last thing you have to bring that is critical to this panel optimization experiment is you need to bring single color cells that are the exact same tissue as your multicolor sample. So you need to have the setup where your multicolor sample and single stain cells are the same in every single way except for the number of antibodies in the tube. That's what we're going to be analyzing in this panel optimization experiment. Now, depending on your setup, you might be using these cells to calculate compensation or unmixing. So you might already have them, but in the event that maybe you're using beads to calculate compensation or unmixing, for this panel optimization experiment, you specifically also need to bring single color cells. And the last point about setup is for those of you who might be doing something a bit larger. I don't want to fully quantify larger. I feel like it depends on your comfort level. So whatever you are comfortable with in terms of panel size and what you feel is a large panel, which you may feel uncomfortable running at this point, I would suggest if you're in that scenario, so I guess I will throw out numbers. If you are currently comfortable with 10 colors and you want to do 20 colors, just as an example, I would recommend optimizing a subset. So just pull out 10 markers. That's what you're comfortable with. Focus fully on optimizing the 10 markers. This is going to be super easy to optimize. And then when you have that nailed down, then you can move up to your full panel. That's going to be the easiest way to make it through this. Now, to analyze this data, you're going to be comparing the single color cells to the multicolor cells and assessing resolution. So it's important to understand that there's actually quite a few factors that impact the overall resolution of your markers. So there's a list on the right there. You can see instrument performance and setup, your antibody titration, antibody performance, whether or not you have high autofluorescent cells or not. If you have appropriately selected a fluorochrome that is bright enough for your marker, and if there's any unmixing or compensation errors. Now you noticed all of these things, this is why we have the readiness criteria. So you need to optimize all of these things when you're setting up before the actual panel optimization, so that when you get to the panel optimization, you know that all of these things are already optimized and you've specifically set up your experiment in a way that we can compare the single stain and the multicolor scenario and assume that any difference in resolution is coming from spread, which would then mean we are assessing our panel. So let's get into some actual data of doing this assessment. I've overlaid single stain cells in the gray line and multicolor cells in the black line. So starting on the left there with our CD45RA, this one actually looks quite good to me. We have pretty similar intensity for the positive population, and the spread in the negative population is the same. So there's no issue with this marker. We're happy with the resolution. In our second example, we can start to notice that the negative population is a little bit wider in the multicolor scenario compared to the single stain scenario. So I am being a little bit more alert now, but the first thing I would do, since histograms can be a little bit challenging to look at, I would first move to a dot plot. And if I look at the dot plot, I can see that I can resolve the CD19 positive cells. So I would say this actually is not really an issue that I'm going to be concerned about. I'm happy with this marker. Then I'm going to move to the next example, CD25. We have the same setup where the width of the negative, the spread in the negative, is a little bit wider in the multicolor scenario compared to the single stain scenario. So this one's a little more complicated. We're going to investigate this one on the next slide. And then the last one, CD127, this one's different. Instead of the negative population, we're seeing differences in the positive population, where the positive population intensity in the multicolor is a little bit dimmer. So this one actually would relate to staining. I would say you would need to investigate your staining in this sense. This wouldn't be related to your panel design. So that since this talk is about panel design, I'm going to focus on the panel design changes, and I'm not going to go into detail about the staining changes. So let's look at CD25, and we are going to figure out what are the next steps we would do to investigate this. So the next step would be you're going to gate every population of interest in your panel, because it may turn out that the source of the spread may not be co-expressed with our markers. So CD25, maybe the spread is coming from cells that are not CD25 positive. However, if you go through and gate out all of your populations and you find that you can't find the populations that you're interested in looking at, then you probably need to change your panel at that point. So doing this with our CD25 example, I just separated these out into individual plots. So we have our multicolor plot at the bottom. You can see there's a lot going on in the negative in terms of spread. So let's change this to a dot plot because it's a little easier to interpret. And in this case, I put the CD25 on the y-axis. So our spread is happening vertically. We can see that there's definitely something going on. But in this case, I did not gate on anything. So this is just all of the cells generally. If I actually went through and gated my population of interest, so in this case, I'm interested in CD25 on my CD3 and CD4 positive cells. And now if I look at that specific subset, I can see that I can actually really nicely resolve my CD25 positive cells. Now this plot looks exactly as I was expecting it to look like. Now, if I want to really investigate where did this come from, I would go back to the SIR matrix. I would look at the column for CD25, all down the column for CD25, look for high value. And I would find that CFLOR BYG610 has a value of 85% compared, so it could potentially reduce the resolution of CD25 quite a bit. But we paired that with CD16. So if we look at CD25 versus CD16, look at that. That's where our spread is coming from. So that's how the SIR matrix is telling us information. But you'll notice that they're not co-expressed. So this was actually a well-designed panel. And so essentially we can ignore this value of 85% in the SIR matrix because these markers are not co-expressed. The last check we can do, if you recalled when we did a theoretical check of your panel and you had some areas where you maybe were a little bit concerned, so we see that value of 95%. Let's check the data. Look at that. We can resolve our CD4 just fine. So even though we had a high SIR value, as long as we were smart about our panel design, we can still get a good panel out of that. So just to wrap up, I talked about a lot of tools and I want to point out just one more. So we have something called our SpectroLearn educational portal. If you thought I went through some of the earlier concepts a lot faster, we have some short videos around 5 to 10 minutes that go into those concepts a little bit more in detail. So feel free to check those out and keep an eye out for new ones that we are releasing. You can find our similarity and complexity index values on our spectrum viewer at spectrum.cytekbio.com. And you can find the panel builder at cloud.cytekbio.com. So quickly summarize, I know we're running out of time. I went through the fundamentals of panel design and how they apply to all cytometers and panels of all sizes. We went through some additional considerations that are appropriate for complex panels and samples that have very high autofluorescence. And we talked about how you can actually run a panel optimization experiment to actually assess marker resolution and figure out if your panel is successful. So with that, I could take a couple questions.
Great. Thank you so much, Laura, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click the Ask a Question box located on the far left of your screen. Now let's get started. So a question here is, do you need to do a panel optimization experiment if you have a small panel?
Yeah, so it's always a good idea to test out your panel before you do a real experiment with some very precious samples. But certainly if you have a very small panel, then it may not be quite as critical. Whereas if you have a larger panel, then it's definitely very critical to do that panel optimization experiment before you run any sort of real experiment.
Okay, thank you. And the next question here is, do you have any pre-designed panels available?
Yeah, so we have a bunch of kits available. And so you can check our website, cytekbio.com, to see what kits we have available. And we're also releasing more in the coming months. And you can also find those kits not only on our website, but they are also in Cytek Cloud. And so even if you wanted to use the kits as a starting place in Cytek Cloud, that's an option for you as well. Or just view them in Cytek Cloud is another option.
Thank you. And we'll wrap with this last question is, do you have a recommended viability dye?
Yeah, so on a spectral system, to give you the maximum number of fluorophores available, I would say live dead blue and Vidi red and zombie NIR are ones that are nice solely because they just don't share any similarity to any fluorochromes that you would want to use for an antibody. But ultimately, you can use whatever viability dye you want, unless you have a highly autofluorescent sample, then I would recommend using something excited by a red laser.
Great. Thank you so much, Laura, for your time today and your important research. We'd also like to thank LabRoots and our sponsor, Cytek Biosciences, for underwriting today's educational webcasts. And before we go, I'd just like to thank the audience for joining us today and for all their interesting questions. All the questions we didn't have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with any one of your colleagues who may have missed today's live event. Until next time, goodbye.